Ladies and gentlemen, good morning or good afternoon, depending on where in the world you're located. I have the pleasure to welcome you to the latest webinar in our series, Scientists Empowering Scientists. We have quite a few people on the call again today. Today we will talk about an open platform 2-photon microscopy, the Sutter MOM 2-photon microscope. My name is Jan Dolzer. I'm one of the product managers here at Sutter Instrument. The agenda today. I will talk about a few introductory remarks, mostly housekeeping items. Then Geoffrey Lambright will talk about the open platform 2-photon microscopy, the Sutter MOM. Aaron Best will host the question and answer session. And then at the end of the webinar, you'll be guided to a quick survey. Our featured product today is the VF1 Edge. The VF1 Edge tunable wavelength switcher. VF, you can imagine that standing for either versachrome filter or variable frequency. What does it do? The VF, the versachrome principle, was a tunable wavelength switcher. The VF1 or the VF5 that is shown here allows you to set the wavelength in a 15 nanometer passband, and somewhere across the spectrum you would do that by defining the center frequency. The VF1 Edge, which we launched last year at Neuroscience, is different in that it allows you to define a passband of variable width, in essence by defining the short pass and the long pass cutoff frequency. So if you're interested in any more information about this product, let us know. Just to let you know, we have it available. With that, let me introduce the speakers, the presenters today. Let me start with Aaron, who will host the Q&A session. He studied at the University of Oklahoma and got his PhD in 2004, went to Harvard Medical School as a postdoc, and since 2011 is one of the product managers at Sutter Instrument. Aaron, as I said, will host the Q&A session at the end of this webinar. Our main speaker is Geoffrey Lambright, who studied at Texas Tech University, got his bachelor in physics in 2001, then went to Germany and in Würzburg made his diploma, the equivalent of a Master's of Science in Physics and Biophysics. Became the imaging facility manager at UCSF in 2006 and 2008, went to Zeiss as an imaging specialist, and since earlier this year, I have the pleasure to call Jeff one of my colleagues. He's one of the product managers here at Sutter Instrument.
Hi, Jan. Thank you. Thank you for the introduction. Hello, everybody. I hope everybody can hear me right now. Welcome to my webinar on Open Platform 2-Photon Microscopy. Today, I will be talking about the Sutter Movable Objective Microscope, aka the MOM. We'll discuss its configurations, the software that runs it, and of course, applications that it performs. So here at Sutter Instrument, we are known for making a wide variety of products, in particular focused for the neuroscience market. However, today what I want to focus on is our microscope division of the company, and in particular discussing the 2-photon microscope, the Sutter MOM, of course. So just to kind of give us an outline of what we're going to be looking at today. First, we'll start off with a quick review on 2-photon technology, how the technique actually works. Then we'll discuss the relevant applications that we can approach with this technique and why 2-photon imaging is important for these fields. Then we'll talk about why a movable objective solution, why the Sutter MOM, and discuss the development of that instrument. And of course, we'll talk about the hardware and the software configuration and flexibility of configuring such an instrument. I'd like to point out right off the bat that it is a highly configurable instrument. If you are looking at purchasing something like this, definitely get in touch with us because we are more than happy to discuss your applications with you and go through the details of really the hardware configuration as well as the software configuration for your particular applications. And finally, we'll finish up with a discussion about advanced techniques and the future of movable objective microscopy. So, for our quick 2-photon review, before I give a 2-photon review, first, I'd actually like to ask everybody in the audience a question. And you will see a poll come up. If you wouldn't mind just answering this, essentially, do you currently use 2-photon imaging in your research? We're just trying to get an idea of how many of you out there are actually using two-photon technology currently, how many are interested and possibly moving into it in the future. So it looks like most of us have voted. Let's go ahead and take a look. And it looks like 39% are actually currently using two-photon technology, and 60% are not, which means that a large portion of you are probably looking at moving into that technology and have questions about the instrumentation. As you have questions, as Jan pointed out, definitely make use of the questions tab in your control panel for the webinar. As he mentioned, all questions will be addressed either at the end during the question and answer session or offline if we get too many questions. Talking about two-photon excitation, in order to understand two-photon excitation, let's first understand one-photon excitation. So what we're looking at here is half of a figure. This figure is showing single photon excitation of a floor. So if we look at this, imagine that we have a floor, and normally the energy level is here at the ground state. However, this reddish purple arrow represents an incoming photon, an excitation photon, that knocks the floor into the excited state. Once the floor is in its excited state, as we all know from our physics lessons, that molecule wants to relax back down into the ground state. So it does so, it comes back down to the ground state, and it has to release the energy as it does so, and we get out a photon, an emission photon, and that's actually the photon that we're observing. So how does two-photon imaging look? If this is the single photon path, two-photon imaging actually happens very similarly, except instead of using a single photon, in the example in the figure shown here, we have a 350 nanometer photon, which relates to that photon's energy. If instead we have two photons of half the energy, double the wavelength, and we hit that floor at exactly the same time with those two half-energy photons, we essentially get the same effect as we had in single photon excitation. In other words, these two photons together hitting our floor can then knock the floor into the excited state. Once the floor is in the excited state, it wants to relax back down to the ground state, and it does so by releasing the same emission photon that we saw in the single photon excitation pathway. So what does that look like? Well, here in this image, we see an image on the left and another image on the right. The image on the left is showing a single photon excitation beam traveling out of the objective above and transversing downward. We see the beam going all the way through this fluorescent medium. We actually see that there is a focal region right here, but we also see above and below, we see a large spread of the beam. In other words, as these photons are coming in, any photon that hits the floor will excite it and cause it to then release an emission photon. And that happens all the way through the axis of the optical path here. However, if we look over at the two-photon side, you'll notice that there is only a small spot right at the focal plane. The reason for this is the IR light, the infrared light, the longer wavelength light of the half-energy photons is not causing any excitation above or below the focal plane. The probability of two photons hitting the floor at exactly the same time can really only happen in the focal plane, and that's why we have the result of excitation only at the focal spot of the laser. So just to clarify, just to make sure we're all on the same page, this is then laser scanning microscopy. So this focal spot is essentially scanned back and forth. And as we scan back and forth line by line, we can raster scan a frame through our sample, through the volume of our sample. And of course, most of the imaging that people are using laser scanning imaging for is to look at three-dimensional structures and recreate our three-dimensional image. So we can then scan that field through the volume. Okay, next I'd like to move on and discuss the relevant applications of two-photon imaging. But before I do, I have one more question for you guys. And in this case, I'm kind of curious what fields people are actually working in. Two-photon imaging is a great advantage for a number of different types of experiments. We're going to talk about a few right now, and it'll be interesting to see where our audience lies. Okay, about 3/4 of us have voted, so let's go ahead and take a look at the results. In basically following my expectation, we have a large community from the neuroscience field. There's a handful of people from immunology and embryology or developmental biology. Of course, there's a chunk of other. And we do have a number of other industrial people online, so those folks are listening in as well. I'm not surprised that we have a large population from the neuroscience community. Again, as I mentioned earlier, Sutter Instruments is pretty widely known for their instrumentation in the neuroscience market. However, I also want to discuss, in particular, immunology and embryology and how two-photon actually helps those fields as well. Okay, so let's move forward. And first, let's discuss neurophysiology. Essentially, two-photon imaging is incredibly important here because you have people are trying to get information out of not just the surface of the brain, but deep into the tissue. The red light or the infrared light of two-photon imaging penetrates the tissue much deeper than the light from single-photon excitation, which is what lends two-photon excitation to be highly valuable for people doing deep tissue imaging. And as well in neuroscience, typically people are trying to get quite high resolution as well as going deep into the tissue. They may be looking at synaptic connections and things like that, as well as the whole cell body. And of course, tracing these neurons deep through tissue can be quite a challenge. So two-photon imaging lends itself as a huge advantage in this field. There's tons and tons of publications on two-photon imaging in neuroscience. I've got 2 examples listed here on this slide that are basically favorites of mine. I'd like to take a look at embryology. For people looking at developmental biology, oftentimes we would like to, say, for example, have an embryo develop over time and be able to image it over and over. However, single photon imaging produces a lot of phototoxicity. And in developmental biology, when we start studying embryos, we find that this phototoxicity actually will alter the behavior or even kill the embryo after very little imaging. For example, if you're using a confocal microscope and you're doing a Z-stack of an embryo, it may be the case that you can only get one good Z-stack out of the embryo before the embryo's behavior has changed or the actual embryo has died. So for people doing those types of experiments, they may find it common practice to say, for example, grow 5 embryos, stop their development at different stages, and then image them, but not be able to image the same embryo over time. However, with two-photon imaging, we do not use the single photon excitation wavelengths. Instead, if we look at our little graphic up here on the right, we use red light or infrared light, and this does not interact with the tissue as much as the single photon excitation light. Therefore, phototoxicity is greatly reduced. And in two-photon imaging in embryology in particular, people find that they can actually image their embryos repeatedly as they develop over time without altering their developmental behavior. And another example in immunology, on this slide I have a picture of a T cell, and one of the reasons I have that image there is because of this first example on the publications that I'd like to share with you. Peter Friedl studies deep tissue T cell migration, and he's done some amazing work in watching T cells migrate through tissue at very impressive depths and at very impressive resolutions. So these are all things that that two-photon imaging has really opened up because of the ability to image deep in the tissue. Of course, in immunology, the lack of phototoxicity is also important for many of the experiments. Okay, let's move forward and talk about why a movable objective solution. So why the Sutter MOM? Well, to discuss that, let's look at a couple of examples of current systems that people have to utilize. One being on the left here, a common two-photon system. and the other on the right, a typical neuroscience rig for slice patch recordings. So here is what I'm using as an example of a common two-photon microscope. This is actually a Sutter instrument. It's a modification to an Olympus BX51. It's a two-photon modification by adding this component that I'm highlighting here. So let's talk a little bit about that. Here is a real-world example. This is an instrument at Duke University. And if we look at this, we notice that the scope is bolted to the table. The sample and manipulators move on a large stage. The whole system is fixed to the table. And a laser is coupled in via a periscope, which is also fixed to the table. So there's no possibility here to move the instrument around because all the optics and the laser is fixed on the table. And there's no room for in vivo specimens, or at least very little room for in vivo specimens, depending on what your specimen is, underneath the objective. On the other side of things, let's take a conventional slice patch rig that many of our neuroscientists are familiar with. And here we have the sample and the manipulators are immobilized on the stage table for stability. The scope and objective translate to access different locations. So if we look at the bottom of the microscope here, we see that the microscope is actually mounted on an XY translator, which means that the microscope itself and even the objective can actually move around the sample area without moving the sample or the manipulators that are attached to the sample. However, because the microscope moves, it cannot be used for two-photon imaging because the two-photon laser would be coupled into the microscope, and then as you translated the microscope optical path would then be off-axis of the optical path of the laser. So that would not work for two-photon imaging. So we developed the Sutter movable objective microscope, which we see here. Just to point out a couple of things, you'll notice the amount of space that is available underneath the microscope. Obviously, that's quite variable as this can move up and down on this rail. On the left-hand side, you'll notice that there is a component here. This is actually the vertical illuminator of an Olympus microscope, and it's actually mounted on its side here. This allows for an interesting wide field path on the microscope. We can attach a camera, eyepieces, filter cubes, fluorescent light source, for example, and do all kinds of standard wide field imaging, if you wished, on your two-photon microscope with the addition of these components. Okay, so how did the Sutter MOM come about? Well, here we go. This is the original prototype. On the left, you see Winfried Dink. He's the actual designer of the MOM microscope. And essentially, he contacted us and said, hey, can I get some of these parts? And essentially, these are three, what you see here is the main components of the Sutter MOM microscope head. 3 single-axis manipulators, which move the optical components in relation to the objective and keep everything in alignment. I'll show you how that works in just a moment. So today, who is this? Who's making this happen? Here is our developmental group at Sutter. Myself, I'm not included in the image, but these are my colleagues, Rick Eyre, Lindsay Dong, and Aaron Best. And moving forward, this is the original patent from Winfried Dink as far as how the optical path is laid out with the mirrors in relation to the objective. And this will make a little bit more sense to you when we see how this works. So this is an actual image of the mirrors. Here is the tube lens that the laser will come out of, and it will bounce off of this first mirror on the left-hand side, go to the mirror that is in the middle of the image, and then bounce down into the objective, which is shown kind of at the bottom of the image. So we can see sort of a schematic, if you can imagine the light path looking something like this. And now what's important to realize is that the objective and this mirror above can translate together, and that would give us our X translation. And you can imagine as this mirror moves left or right, it's still going to reflect the same beam down into the objective. And that's kind of the key idea behind the whole microscope head of this instrument. Furthermore, the objective can be moved in relation to this mirror to give us Z translation, which is actually our focus. And if you imagine these arrows are actually intended to be in and out of the frame of reference here, so in and out of the screen. So in other words, these three components can all move back and forth together, which would then give us our Y translation. So one of the nifty things about this design, as we can see in the patent, is that rotation is also possible around the optical axis. So what does that all look like? So this is a video of the actual microscope head, and I'd like to just show you so you can imagine how this works. Now we see the microscope is actually translating in X right now, just to give you an idea of how this operates. But as I mentioned, rotation is also possible around the optical axis. And this allows for imaging non-orthogonal surfaces to the microscope, to the table surface, for example. And you can take that to the extreme level and simply convert your upright microscope to an inverted microscope. This might be important for embryologists, for example, people that are looking at things developing in a petri dish over time. So the flexibility of this microscope head is quite a convenience for a number of samples, as you can imagine. Okay, so next let's talk a little bit about the hardware and the software options for this microscope. First of all, the scanners for the laser scanning. We can incorporate galvanometric or resonant scanners. Galvanometric scanners are important for folks that, for people that want to do highly precise positioning of the image area. For example, if you want to go in and scan only a certain region, or maybe a couple of regions, some people call this postal stamp scanning. Resonant scanners, on the other hand, are very, very fast, but you don't have the precise control that you have with a galvanometric scanner. So you get fast full frame imaging with a resonant scanner. Laser options, of course, the laser is one of the most important components of the two-photon microscope. On the left, we see spectrophysics lasers. On the right, we see coherent lasers. Those are the two most common options for most of our two-photon customers. However, there are some other emerging laser manufacturers making pulsed titanium sapphire lasers, which are required for two-photon excitation. However, it's also important to note that you do have additional beam access, you could say, with this microscope, and I'll show some of the options there. But for things like photostimulation or reflection imaging, virtually any laser can be aligned into this microscope. Okay, we also have a variety of detector options. We could incorporate standard PMTs. These are photomultiplier tubes, the bread and butter of confocal detectors. We can also incorporate gallium arsenide phosphide PMTs, which are highly sensitive. There are also gated PMT options for folks, for people that are doing photostimulation. For example, they might have a very intense pulse of stimulation light, and they want to gate their PMT off during that stimulation pulse in order to protect the PMT. There are also PMTs for photon counting, which leads to things like lifetime imaging, for example. Okay, and how can we mount those detectors on our microscope? Well, here are three detector arrangements. On the left-hand side, we see a two-detector option. One PMT would fit right here, and the other PMT would be in this module right here. Filtering done here and here. And what you'll notice is that the objective is down below, just underneath this, and the image, or the emission light comes up through the tube and to the detectors. What's nice about this arrangement is that it leaves a lot of room open around the objective. We also have a 4-channel version of this. So here you see a 4-channel version, and otherwise it's the same thing. On the right-hand side, however, we have what's called a short path. You'll notice here that the emission light comes out of the objective and straight over to the PMT module. This is a shorter light path, making it a little bit more efficient for photon collection. And there's one other option that's not shown here, and that's a wide path actually, which would be this version with larger optics for more photon collection efficiency. So a number of options here, depending on whether or not you need a lot of space access under your microscope, or if you're looking at very, very dim things and just trying to squeeze out every bit of signal that you can. All right. Of course, there's also a variety of available objectives in the world, you could say. And indeed, the Sutter MOM microscope can incorporate any of those objectives. What's important to remember is that the color corrections that are done in the objective, the objectives used in the microscope should be fully color corrected for optimal performance. Okay, I mentioned a moment ago the idea of accessing the beam path. This is one of the things that makes the Sutter MOM microscope attractive to many users is the idea of customization. So in this particular case, what we're looking at is a portion of the beam path. You actually see the scan lens right here very prominently. And so if you look just to the right of the scan lens, you'll notice the two scanning mirrors. Of course, we have the beam path is open and uncovered here, so I can show it to you. And the beam would go from the scan mirrors through the scan lens and then forward into here, and then it's not shown, but a tube lens is inside this module. So here you can visualize the beam path, what the beam path would look like. And what I'd like to show you is actually this area right here. So I've just put a picture here of this white cube. In this particular case, this example, this is a polarization beam combining cube. And so now you can imagine that we can run our IR beam, our pulsed infrared beam for 2-photon imaging through this cube and onto the sample. But we can also bring in a secondary beam and combine it with that beam and deliver that to the sample as well. Just a note, this beam could be used for a stimulation pulse, for example, or you could even have an entirely separate set of scanners and scan lens and independently scan this beam as well as this beam. All right, so with all those hardware options, how do we control all of this? With essentially any detector, any laser, any of these options, it turns out we have a number of software options as well. The two primary software options are Sutter MScan and then Scan Image, which is supported by a company called Vidrio. There are also other options. For example, people have built their own software utilizing MATLAB, LabVIEW in order to make their own custom software. There's also a few packages out there called MPscope, Helioscan, and I think there's a couple of others that I may be less familiar with. But as I mentioned, the two primary software packages are Sutter MScan and Scan Image. So I'd like to tell you just a little bit more about those. The Sutter MScan software is designed to be relatively intuitive, easy to approach, good for a new user especially. It's fully integrated with the system, and if you're looking for a turnkey 2-photon microscope, the Sutter MScan software is probably your best option, as it will be fully integrated in the computer, and it's really easy to use and approach. However, it turns out for most of our neuroscientists, there's a program called Scan Image, which if you do 2-photon imaging and you're a neuroscientist, you're probably already familiar with Scan Image. Scan Image is a neurophysiology focused, very, very powerful software. Its biggest benefit, well, there's two great benefits for neuroscience here. It's highly integratable with other equipment. So a lot of these electrical recording experiments and things like that can be kind of networked to this software, so to speak, so that you can control your other equipment in tandem with the actual imaging platform as well. Okay. So moving forward, we're going to discuss advanced techniques and the future of MOM. So one of the things I'd like to talk about a little bit is I want to discuss the idea of 3-photon imaging. This is something that's been theorized and maybe practiced a little bit in the past, but due to technological reasons, it's been limited in its performance so far. However, in the last few years, there's a group, Chris Xu, who has made some pretty big advancements here. Let's just take a step back and talk about our Jablonski diagram, which we discussed earlier. We remember single photon excitation, the idea that we can knock a fluorophore into the excited state with a photon. And of course, two photon excitation, we take two photons of half the energy and hit that fluorophore at exactly the same time. But what if we extend this graph a little bit, and instead of two photons, we take one, two, and then three. So in other words, we're going to take three photons of a third of the energy each, and try to take those three photons and hit the molecule at exactly the same time. So the effect would be similar to two photon imaging. However, the excitation wavelengths would need to be much longer. And that's why this is starting to become more of an approachable process now, is because lasers are actually being developed now, something called soliton lasers, that are actually providing wavelengths out in this range with enough power to get this imaging done. So I highly recommend, I'm not able to show the data from this publication due to copyright reasons, but I highly recommend looking, if you're interested in three photon microscopy at all, looking at some of their data. The image quality that's coming out of this is pretty fantastic. It's not completely intuitive why, but as you look at it, there's some good reasons why three photon imaging will provide some good results. All right, I also want to discuss a little bit more about the flexibility of this system. So here we are, we're looking at the Sutter MOM. I had mentioned that we have access to the beam path in a number of ways, so I just kind of like to point out some of that flexibility right now. I've got a little red arrow here drawn, which shows our infrared beam coming in. It would come into the periscope and go up to the microscope head for two photon imaging. And I also, a moment ago, showed you a beam combining cube that could be used to input another beam in the side. So you have that access as well. However, you can also access the beam path behind the scan lens. So you can bring a beam in between the scan lenses between the scanning mirrors through the scan lens, and then on through the tube lens and into the objective as well. And then don't forget, we've got this whole wide field path over here with the flexibility of all of the filter cubes and the wide field imaging path here. We could also bring in a laser beam through that wide field path if we wanted to. And indeed, here's a group and a paper that publishes a very nice schematic actually of how they're bringing in a stimulation beam through the wide field path on our system. All right. So with that image in mind of all these different access points of the microscope, it's important to remember not only can it be a turnkey system that's up and ready to run, but you can also then tinker and build and add components, as many of our customers are doing with their systems, and start to tackle a whole wide range of various technology and applications here. All right. With that being said, I'd like to open us up to questions. So just a reminder, please do use your questions tab on the left-hand side. At this point, I'll welcome my colleague Aaron Best as well.
Hi, Jeff.
Hi, Aaron. How are you?
Good. How are you?
Good.
So we have a number of questions already, actually.
Great.
So I think I'll just go ahead and jump in.
Yeah, if you don't mind, you can address the questions directly, and I can help out either way. Why don't you go ahead and read some of the interesting ones that pop up.
Okay, we can talk about them together. So the first question is, this user wants to use a MOM for 2-photon FRAP. I want to photo bleach a small volume for only a very short time at high laser power. Is there a mode to photo bleach only a small volume at high laser power and rapidly change to normal laser intensity? And one of the complications the user indicates is that they only want to use one Ti-Sapphire laser and one Pockle cell to do this.
Oh. Well, if you're limiting it to one laser and one Pockle cell, and feel free to add your comments on top of this as well. it seems to me that you would have to be able to control that laser. Essentially, you would have to use the same laser for imaging as well as for the photo bleaching portion of the experiment. So what would have to happen is you would scan a whole area at imaging power. Then you would potentially zoom into an area that you want to photo bleach. So in that case, I would think galvo scanners would be really key to making that possible. In other words, with galvo scanners, you can actually control where the beam is going. So essentially, you would image your full frame, use the galvo scanner to point to your frap region, essentially, turn up the power from the Pockle cell, in other words, basically turning up the laser power, and then scan that area to frap it or bleach it, and then go back to the imaging power in the full frame and watch the recovery in that way.
And I think one of the keys will be the fact that Pockle cells can modulate beam power at very, very high rates. And so asking about specifically being able to go back and forth between different power settings, I think that's the key.
Right. Yeah, the Pockle cell would not be the limitation there. It would be how quick you could essentially scan a whole field, zoom in, scan a region, zoom back out, and scan the whole field again. So
correct.
The scanning would be the limitation in that sense.
Yes.
Just to note on that, actually, there's a number of growing requests to incorporate a second scanner. In fact, that little beam combining cube and two pathways that I was showing, where I had mentioned that you could have a second set of scanners, for example, input in that area. A lot of people are doing experiments like this where they want to, for example, with the Thai sapphire laser, image the whole field of view constantly, and then separately go into a certain region and, in the case of this example question, photo bleach an area. But many people are doing things like photo stimulation as well. And so they might use that second set of scanners to independently control their photo stimulation beam while the first set of scanners is continuing the imaging.
So our next question is, does the field of view of the microscope change with objective positions? More specifically, at the extremes of travel, is the field of view clipped?
No.
Yeah, that's, sorry, fairly short answer, but that's all you need to say. Okay. The next question is actually about detector heads. And so we have a number of different detector heads, and sometimes it's difficult for people to know which is the most appropriate for their particular application. And this user is asking, what are the advantages or disadvantages of the short versus long detector path layouts?
You want to go for it?
Sure. So the biggest difference between the different detectors is whether or not they have a mirror immediately to the right of the main dichroic that collects the emitted photons. And the reason that a number of them are designed to have a mirror that then reflects the emitted photons up is to actually provide access near the objective for pipettes mounted on micro positioners. And so you're adding distance to the actual collection pathway, which could produce a decrease in collection efficiency. But the reason is because you need pipette access, which is very, very common in the experiments. Now, then the question becomes, how much of a deficit is it to add that? And I'd say it probably depends. It depends on how deep you're imaging, how large your back aperture of your objective is, the level of scattering induced by your particular sample. We have done some tests. I ran some tests at Berkeley comparing the short path detector to the standard Pentagon detectors. And if you basically use the same filters in the two detectors and used a relatively small back aperture objective, an Olympus 40x 0.8 NA objective, there was actually very little difference. Now, if we were to use something like a Nikon 16x objective and image very deep, that might have been different. Now, I should say one of the objectives that wasn't shown is our wide path detector, and this is also now known as the Janelia detector because it was designed by a group at Janelia Farms. And this detector was actually designed specifically to get over some of these shortcomings, to try to get collection efficiency up with these large back aperture objectives, but still give access to the preparation. And so it has a mirror immediately to the right of that main dichroic, but the collection, the detector, the entire metal structure is of larger diameter. So it's 50 millimeters in diameter up to the point at which the two colors of the emitted photons are separated, and after that point it's 30 millimeters in diameter. So that's generally a good compromise, and it's probably our most commonly purchased detector. Okay, so this next question is, can I ask where the scanning mirror is located in the MOM, and is a galvo mirror or resonance scanner integrated into the MOM?
So I pointed out on the slide, and we can go back to it, showing where the scanning. I'm not quite sure what they want to know. Maybe they can offer a little bit of clarification on the question.
So I guess what I would say is that the optical axis of the MOM is running horizontally from front to back. So the scanner location, the primary scanner location, is on the back of the microscope on the right, and then it enters the scan lens and tube lens along a vertical path. So that's the actual location, and then a periscope brings the laser up off the table into those mirrors. The question was, is a galvo mirror or resonance scanner integrated in the MOM? And it's actually, you can have either one, or we're designing hardware to actually include both if you need two separate sets of scanners. But both are very common. Check to see if there's any more questions. So Jeff, one of the things that I was very curious about was the increase in the use of 3-photon imaging. This is something that's really fascinating, but I always assumed that it wouldn't be very useful because of the very, very low probability of getting 3 photons absorbed in a fluorophore within the focal volume within a sample. What is it that's making this possible now?
So it's something I'm excited about. So I'm glad you're bringing it up. I kind of felt the same way when I've talked about 3-photon imaging in the past. It's something that it sounds like you're taking all of the challenges of 2-photon imaging and magnifying them. Essentially, the cross sections, the excitation cross sections of the fluorophore change at between single photon, 2-photon, and 3-photon. And basically, what it means is that as you get more and more complex, there it becomes harder and harder to excite the fluorophores. So you're dealing with not only the probability and the difficulty of the probability of getting 3 photons to hit at the same time, but you're also dealing with relatively low excitation efficiencies as well. However, that being said, I think the biggest thing that's prevented people from doing 3-photon excitation is having enough laser power out in the IR range where this could be really important. And what we've seen in the last decade to half of a decade is the development of what's called soliton lasers. Unfortunately, I don't know too much about this technology, but essentially what they're able to do is produce a lot of laser power at various wavelengths like 13, 1400, 1500, 1700 nanometers, things like that. And that reference I showed earlier from Chris Hsu, indeed, they're actually doing their work at 1700 nanometers. So what that means is that actually you can avoid one of the biggest problems that you have with 2-photon imaging, and that is the absorption of water in the near IR wavelengths. So water has a lot of absorption peaks in 850, 900, 950 in this kind of range, which is very common range for doing 2-photon imaging. So typically what limits the amount of power that you can supply to a sample is not photo bleaching. For example, when you go onto a normal confocal, you can basically crank up the laser until you start to photo bleach. With two-photon imaging, it's a little bit different. When that water absorbs the IR light, it heats up. It actually boils. And so any of you that are familiar with two-photon imaging, you're probably used to adjusting your laser power, trying to squeeze a little bit more laser power into the sample so that you can squeeze a little more emission light out of the sample, and then suddenly the sample literally explodes right in front of you. And so that's one of the challenges of two-photon imaging. Now with three-photon imaging, getting out to 1700 nanometers, you actually avoid a lot of those water absorption peaks. And what that means is that you're able to really start cranking up the laser power even more. So that 1700 nanometer light is not interacting with the tissue, but you can get enough power to go through that you actually have the probability of 3 photons hitting the floor at exactly the same time in that same space in the focal volume.
Thank you, Jeff. That's
Sure.
great. Thank you.
And I have to say, I mean, the image quality is pretty darn cool. It's neat what people are getting.
Yeah, it's really cool, definitely. Now, additionally, someone asked a question I think is pretty interesting. So what about substage detectors? If you use the MOM with brain slices, for instance, as opposed to in vivo experiments, could you increase detection efficiency?
Yes, and actually who it's more important for are people that are doing experiments with collagen in particular. So collagen has an interesting effect when you hit it with IR light. It's not two-photon excitation, but for those that are familiar with the field, second harmonic generation of the collagen fibers actually is created with essentially a two-photon beam. The interesting thing about second harmonic generation as opposed to fluorescence, fluorescence, once a molecule is excited, it emits light in all directions. You know, if you imagine the sphere, it's coming out below, above, left, right, behind, and in front. However, second harmonic generation is actually a very forward process. There is a back signal in second harmonic generation, but about 80% of the signal from second harmonic generation goes forward. And indeed, having a detector essentially in front of the beam on the transmission side would hugely help for second harmonic generation. It can be used for a little extra fluorescence collection as well.
Yeah, so actually a couple of other details. So we have had some people add substage detectors to the MOM. It's a little bit difficult actually because you have to coordinate their movement with the positioning of the head of the MOM unless you want to actually just set the MOM in a specific location and not move the slides. But there are actually real advantages to substage detectors. So one thing that can be done is you can actually use a condenser with a higher numerical aperture than the one that's actually than the numerical aperture of the objective. So at times you can actually get more collection substage than you do above. Actually, and this is why we have substage detectors as an option on our more slice-oriented two-photon microscope, the DF scope kit for making BX51WI microscopes into a two-photon. Because I think that's a very useful bit of information for people to know that if they need more signal, a lot of times they can get that by collecting from below. So this person would like to know, getting back to the question of switching between full scan of an image and bleaching a point of light, 2 issues. Which software has this capability, and how quickly can a laser change wavelength?
Okay, so in the FRAP experiment, we wouldn't change the wavelength of the laser, we would only change the power of the laser in order to bleach the molecules. If you're trying to change the wavelength of the laser, for example, some people will scan a Z-stack, change the laser wavelength, and then scan that Z-stack again. It depends on the lasers, but generally that's a relatively slow process. For example, if you're changing from 800 to 900 nanometers, that would take on the order of a second, a couple of seconds, somewhere in that range, depending on the laser, of course. So you generally don't want to do laser wavelength changing during your experiment because if you have to repeat that, those seconds add up pretty quickly. However, that's when people are combining 2 lasers, for example, and bringing them into the microscope together. They would have one laser tuned at 800 nanometers, for example, and the other laser maybe at 980, and then bring those in and either scan them simultaneously or sequentially depending on their application.
So this same user was wondering which software has the capability to do this. Now, MScan has the ability to use 2 different laser light sources coming through the same set of scan mirrors running through 2 separate buccal cells. So if your particular photostimulation scenario is amenable to those details, the MScan would work. ScanImage has a number of abilities with regard to photostimulation, and it's probably best to ask specifically what the end user needs before trying to explain those different capabilities.
Typically an experiment like that would be set up as kind of a time lapse where you have a certain number of images, and then you have an event, or that event can be zoom in and pulse something, or zoom in and image this little region, and then continue the rest of the frames of the time lapse back with the settings of the first 3 images, or something like that. I can't speak for Vidrio on the functionality of ScanImage, but with my limited experience with ScanImage, I would think that they probably may have that functionality in the software. I'm going to be very careful with my wording there. Maybe if there's somebody from Vidrio on, they could chime in.
Okay, I think this is a good point to wrap this up for today. We're almost an hour, and still have most of the audience there. I would like to thank everybody for being here today, for a very interesting discussion. A lot of questions, a lot of interesting, important questions were asked. Remember, you have the opportunity in the survey that you routed to after the webinar to ask any further questions. You can also ask us to get in touch with you if you have unrelated support questions or anything. Please let us know how we did, how you liked it, whether it was useful for you. That is important feedback for us to know what we can improve going forward. Thanks again for being here. We'll leave this open for another couple minutes, so if somebody's still typing a question, they can finish that and then close the session. Stay tuned for the email notification about the video, as I said, probably within the next 2 weeks. Thanks a lot again for being here, and have a wonderful day.